Okay, so this is just a quick run through of the mark scheme and any issues I had. I don't think I'm going to go through every single question, so anything that I got correct, I'm just going to skip. I'm not going to discuss that, but I will highlight where I lost marks so that whatever my thought process was in terms of answering that question, clearly that was wrong in some way, so we will have a look at those bits in particular. So I've highlighted them in the paper where I lost marks. So first question, I know, a little bit embarrassing, but that's fine. Okay, so question 1B, part 1. What we had, I'll focus on the main parts of the mark scheme. So what I forgot to say was behind the endothelium for some reason. Allow artery wall, atheroma formation can develop into fibrous plaque now. Yeah, so I just forgot to mention, or I didn't mention, the artery wall or even behind the endothelium of the artery. So yeah, that's a bit disappointing, but that's fair enough. Okay, and then most of the other parts were okay. So generally speaking, I did find this question paper a bit tricky to deal with, but the mark schemes are kind of more or less in line with what I was thinking, apart from this one. So the interferon question, that's C. What question was that? Question 3C. So let's go to question 3C. 3C is where I lost a lot of marks. So we'll zoom in on the main part of the mark scheme, because that's where you really want to be working. Okay, so explain the results. So increasing dose of interferon increases the survival time of mice. So I got that mark describing the data. Fine. Because interferon inhibits viral replication, allow interferon prevents virus infecting other cells. So I really didn't do a good job of reduce rate of infection of mice cells. So maybe I would give myself that mark there based on the allowing stuff. So based on that, allow interferon prevents virus infecting other cells. I might give myself this, reduce the rate of infection of mice cells. Okay, thank you. By influenza virus. So maybe I got a 2 out of 3 for this one. But I didn't say this part, which is disappointing. Well, maybe I get marking point 2. The greater the dose of interferon, the fewer virus particles produced. So yeah, didn't really say that. Interferon prevents virus entry into uninfected cells. The greater doses of interferon would reduce the, yeah. So I said what it does, but then I didn't make a comment about the increasing dose and the increasing effect. Okay, so that's fine. That's fine. I'm not too disappointed with that one. Right, let's look at the next highlighted bit. Okay, so here describe the role of tRNA, question 4. So let's go to question 4. All in all, I think fair mark scheme. But sometimes it does seem a little bit like if you're only going to have 3 marking points, I think that's a little bit, because there's so much, there's quite a lot to talk about in terms of what tRNA do. But yeah, I didn't say each tRNA carries a particular amino acid. I did say they bind specific amino acids, so I did say that, but maybe I didn't say that each tRNA carries a particular amino acid. So, okay, missed that mark. Fair enough, fair enough. And it is an important part of what they are doing, the tRNA. Okay, right, next one. This was very disappointing. Even though I was thinking globular proteins, I didn't include that in my answer. So there's a lesson for everyone here, including me. if you got a multi-mark question, like 3 marks or above, or actually any question, I think there's the first sentence you say has to be the most obvious thing. And because I think I've lost quite a few marks for not saying obvious things. So yeah, so let's just go to that mark scheme. Okay, part 3. So primary structure determines the folding of the polypeptide. Primary structure is a sequence of amino acid. Tertiary structure is formed. Okay. Water. So I think the marks I missed here were this one and this one. Yeah, I don't know. I don't know if water is technically forming hydrogen bonds with hydrophilic groups. I mean, it could be, but I don't always think like that. Dipole-dipole interactions is fine. Hydrophilic interactions? Okay, I will remember that for the future. That we do need to describe the interaction that water has with these molecules, not just saying that water has an interaction. Okay, learned my lesson. Let's look at the next place where I lost marks. Okay, stem cells. So what question was that? That was question 5. Question 5, 5b, part 1. Okay, so state what is meant by the term stem cell. I mean, I got undifferentiated cells fine, but yeah, you know what, this is fair enough. A cell that can give rise to specialized cells. So I think sometimes, yeah, we can fall into the trap of saying that I said undifferentiated cells. However, stem cells don't, stem cells don't become differentiated. They kind of divide, and then the cells that are produced from that division, it's those that become specialized. So it's not fair. Maybe it's not technically true to say that stem cells become specialized. Maybe we should say stem cells give rise to specialized cells, which is fine. I'm okay with that. And yeah, the two things about stem cells is that they can give rise to specialized cells, and they can divide many times. So I said unlimited replicative potential, so maybe I give myself that mark. However, I see this term Hayflick limit come up a lot, and so I think I need to train myself to maybe say this, Hayflick limit. Okay, next part. Oh, lost 2 marks on this one. Yeah, because, why did I lose marks on this one? So compare and contrast the results of mitosis and meiosis. So I think I understood the question fine. The results, I did address the question of, okay, it's not just meiosis versus mitosis, it's about comparing the products of those processes. But maybe I didn't quite think deeply enough to give the answers here. What did I miss here? Let's see. I think I didn't say the super obvious thing, which is they both increase the number of cells. Yeah, that was dumb. And then, in terms of mitosis produces diploid cells, I got the second marking point. Meiosis produces, and I got the third marking point, but I didn't comment on the number of cells. So both increase the number of cells, but one increases to a certain number and the other one increases to a different number. So yeah, I should have at least thought of the 2 and the 4 cells, because mitosis is 1 division and it results in 2 cells. Meiosis is technically 2 nuclear divisions, and so or 2 cell divisions, and so it produces 4 cells. I really should have thought of that. So yeah. Do excuse my handwriting. I mean, I would advise you to write better than I am. It's just I'm using a small iPad, and so my handwriting doesn't come out necessarily as it would on the page. At least I have that excuse, and Yeah. Okay, question 6. That was fine. I'm always reluctant to give 6 marks on these questions. But I think, the criticism I would give myself in this answer is that from the mark scheme, so let's go over here. From the mark scheme, what you want to try and do is get a couple points from here and a couple points from here and at least a couple points from there. That balance is what can result in getting the higher levels here. So I've stopped short of giving myself the full marks because while I got marks from this area and this area, I only got maybe 1 point from the last section. So I need to be a little bit more observant of the information and spot a few more patterns. Just saying a few more, because comment is really like, what do you notice? And maybe here I just didn't notice enough stuff. Okay. Okay, let's move on. Okay, this question was tricky to work through, but in the end, turns out okay. I missed marking point 5 on this question, so that's question 7, right? So question 7, 5 marker. The change in the range, so I forgot to be specific, or I didn't think about being specific about what could be the causes of the change in the pathogen of the ash trees linked to a relevant aspect of climate change. Yeah, so this is a big part of topic 5, like how global warming has its effect. It's not just temperature well, actually, I did say temperature increase. Adapted increase in atmospheric temperature. So I did say that. So maybe I did get all the marks there. Because yeah, I think I did get that mark. All right, question 8 was mostly okay. Molecular phylogeny, I think, was fine. And then that was all right. Yeah, this one, I just got that wrong. I just basically got that wrong, and it's not the first time I've got this one wrong, actually. Let's see, question 8. Right, question 8, and we're looking for which concentration and why. So this kind of question comes up a lot, so I'm going to spend a little bit of time here. Now, this is really important to understand. Explain which range of enzyme concentration should be used to compare the activity of trypsin. So what you want is a range where it's only the enzyme concentration that's really affecting the rate. So if we look at this graph, the part of the graph, so my logic was fine, but also a little bit not fine. So I was basing my answer on just the steepness of the curve. And while that's okay, I think I was missing a key point, which is while the best range is at the lower end of the concentrations, i.e. up to 2, and I only went up to, I think, 1, or 0.1, or 1, actually, I think the key thing here was, look what happens to the curve after that concentration. It levels off, which means enzyme concentration is no longer a limiting factor. So if we're investigating enzyme concentration, we want the enzyme concentration to be the limiting factor. So what I should have noticed is that because there's a leveling off above 2, anything below 2 is okay, because below 2 the enzyme concentration is the limiting factor. Above 2, something else has become the limiting factor. Yeah? So, yeah. Okay, and I don't know if I would have said this, but initial rates of reaction have to be compared. Okay, I don't know if I would have said that. Right, then 9 was okay. So I'm only skipping over these parts because you can see my thought process for these questions, and it seems like my approach was correct, so you can just watch that. But I'm making this kind of add-on so that you can see all the bits where I went wrong. I don't know what I did here. I got this part of the calculation correct, but for some reason I've ended up, I've either written the wrong number or I pressed the wrong buttons on the calculator for whatever reason, maybe missed a 0, but I have ended up with an answer that is out by a factor of 10. So, yeah. And maybe I should have noticed that because, well, no, actually, no, I don't know what's gone wrong there. I really don't. 60 millimeters becomes 60,000 micrometers. That seems all right. Okay. Yeah, on this question with the photosynthesis, so still question 10, I missed a mark because I talked about, so my approach was correct. My approach was right. So pick a structure and say what its role is in photosynthesis. But for the last one, I said the electron, because for 3 marks, I didn't know whether I needed to go to that level of detail. I thought that might have been a bit extra, but I had to say that the electron carrier molecules are, that their role is in ATP production or photophosphorylation. That would have been fine. But I just didn't say that. I just didn't go that little extra bit. I said like dependent stage, but not specifically ATP production or photophosphorylation. Okay. Yeah, here I tripped up a little bit because I called the intermediate GP rather than GALP or TP. Yeah, that was just a error. But subsequently, after that, I did get the marks. So, but yeah, I did lose a mark for kind of making that mistake there. So that was supposed to be GALP or TP. Then, yeah, and then the last question was it a 6 marker. So I left this kind of in a planning stage. I didn't really write everything down, but I've put all the main points down. Now let's just go to the mark scheme. What did I miss? Compare the time, measure the effectiveness. I think, yeah, I did. Why did I give myself 5? Maybe I just gave myself 5 because leaving that answer in that kind of state doesn't deserve 6 marks. That's just how it is. But I think I talked about all 4 herbicides. I did talk about controlling light intensity, so I got a couple from that part. Sensible herbicide. Now I didn't talk about herbicide concentrations, and you really should do that because they do highlight the concentrations here, right? So using those as part of your method is always good practice. I did talk about isolating chloroplasts and controlling abiotic variables. I did talk about using DCPIP, like how would we know which herbicide had the biggest effect? Did talk about comparisons. I didn't talk about tubes in the dark. I did talk about how we're going to quantify using a colorimeter. So maybe this is bordering 5/6, but yeah. Do I want to be that guy who's giving himself 6 out of 6? Maybe. Yeah, but I think, look, should I have said a few more things? Yes, and if that's the case, I'm not going to give myself 6 marks. Okay, and that's it. That's the 2018 paper 1. I'll see you for the next one, I guess, hopefully. Hopefully you're finding these videos useful. If there's any kind of feedback, any improvements I could make to make the videos a bit more useful, please do let me know.